Okay, so we had a lively discussion on the computational issues. As every discussion on the computational issues, we had to start with this. I think, let's see if I have a pointer here. So actually, initially when framing the questions, we expected to focus on the portal side of things, on basically a variety of tools that people would like to create or would like to see that would make use of the HCA data and information there. And in fact, there's actually quite a few initiatives already in this space solicited by CZI. This is kind of an overview. Thank you to Jeremy and CZI to put this together nicely. Overview of different portal projects that are, I think, about to be funded, just to show you the scope of these portals that people are proposing. And they vary from sort of very technical things, for instance, creating the right APIs, computational modes for making use of or representing this data, processing sort of new cell types, allowing to scale up calculations, to sort of novel analysis types, making use of, let's say, population variation, bringing this in and comparing with the HCA data. Also refining the actual interpretation of single cell data, let's say with trajectory, cell type, cell state definitions, and so forth. So these are the types of portals that we expect people to be creating, and I'm sure there'll be a lot more ideas along these lines coming forward. And so this is sort of the plan that we had. In fact, I think the discussion ended up being mostly centered on creation of the portal and what we would like to see in the portal itself. So as I said, the aim was to try to solicit community input as to what were major computational challenges that are facing us. We wanted to go through these use cases as far as portals are concerned, but also important use cases for actually contributing data to the HCA. The annotation I'll discuss in detail ended up being a major topic there, and we also wanted to contribute some time to discussing technical issues related to actual creation of the portals and maintenance of that data. Okay, so this the annotation and interpretation actually ended up being a topic I think we spent the vast majority of the time on. So the simple way to fall into this is to come up with a basic portal, a cell blast portal. So let's say you come in with an external data set and you would like to basically use HCA data to help you annotate and make sense of what you're seeing in your own experiment. And there are actually a few tools already that attempt to do this, CMAP and RCA. As an example, in terms of algorithms that could potentially do this. And based on such algorithms, you could build a portal that would actually perform this. So from the sort of user standpoint, it was brought up that it's important, not just to assign a label, for instance, but also provide, some measure of confidence as how confident we are in let's say that this particular cell cluster is let's say astrocytes. Algorithms make mistakes. They sometimes can estimate the probability with which they'll make mistakes, so we can relate that information. Obviously, people make mistakes too, and that has to do more with the creation of these annotations. And so we would like to be able to provide sort of evidence. I think an important point was made by Chris that we have to separate prior knowledge and inferred knowledge. And that goes back to sort of reporting confidence in different levels of evidence. There was a discussion about, in terms of annotations and interpretation of the data, what levels of interpretation we'd like to see. Obviously, the simplest case, you could just have a simple label, very succinct. You could provide evidence, for instance, as a list of marker genes or sort of very specific signatures that are supporting this type of definition that the annotation ended up being. Now, the marker panels obviously don't work in all the cases and sometimes oversimplify things. So it's important to keep in mind sort of the level of detail that the user would expect. And we expect different types of users from sort of biologists who are not working with intimately with single cell data, and they would just like to see the most succinct interpretation, to bioinformaticians and specialists in that particular tissue would like to see a very extensive list of evidence. And I think both will have to be somehow provided by these, both levels would have to be provided by these portals eventually. So relating to all this, or underlying all this, are the issues related to creation of the annotation, which has to do, I guess, with the creation of the atlas itself. Well, the first thing that was pointed out is that we should try to be more formal and in terms of terminology we use for describing, let's say, cell properties. And cell ontology, or some kind of ontology that we'll be using, that we should be all agreed upon and using in this effort. There are some ontology efforts already ongoing, especially here at the EBI, for describing this, and I think there was kind of community enthusiasm about trying to refine them further and bringing them into this annotation process. Also, when we talk about tracking evidence, so whenever some kind of manual annotations or input are made, we would like to be able to keep track of evidence. We would like to have an evidence trail. And related to this, you can actually have ontology of evidence, which has been used in other projects and can be very useful for then systematically assessing or filtering, certain types of annotations that will be more confident. Again, I think the challenge was reflected well in this discussion, was also plurality of opinions as to what the exact annotation should look like. But in the specific cases, in the specific data sets, we'll also see multiple experts annotating things differently. And so I think for now we'll basically, it's important to keep track of it, of different opinions, and then we can try to resolve them later. There was an important point that we should also be annotating unwanted factors. We'll talk about batch correction in a second, but if you have, let's say, some kind of a technical effect affecting a particular data set or cells, let's say heat shock signature and so on, we would actually like to have this annotated, both in general across data sets and for specific instances. An important point was made that we'd like to use this very high-dimensional data to create very refined, very fine clusters, very detailed annotations and subdivisions of cells. And at the end, they will have to be validated somehow. So some discussion was there about what types of evidence can we start bringing in or collecting eventually to support creation of new subpopulations and so on. And HCA could potentially serve as a central hub for coordinating this, keeping track of that information. Sorry. Use cases. So this is actually what we thought, in terms of these red box portals, we would spend most of the time on. And a few were mentioned. So one fairly basic but obviously very important use case is an overview of just cell frequency. So in the most basic case, having some kind of a portal that would simply show distribution of cell type, cell state frequencies across collection, across different tissues, let's say within a tissue. But that could also be used to perform more detailed analysis, such as analysis of shifts of populations between different conditions or between different samples or tissues. And so this is kind of an example of more of a meta-analysis portal that wouldn't necessarily reanalyze the expression data itself, but would use the summary statistics from the HCA to run additional analysis. So another use case, another use case was integrating HCA data with GWAS. And try to make use of the single cell data. Sorry. Trying to make use of single cell expression data to refine GWAS analysis, GWAS results. There was a suggestion to, again, abstract the results of the HCA data set to sort of create metacells. Which would be more abstract descriptions of cell type, cell states that could be quantitatively described and then served as sort of input to additional analysis in other contexts. There were suggestions for obviously integrating HCA with other data, so there could be separate portals, for instance, showing showing how different HCA cell types and their combinations would map to or correspond to data collected by, let's say, ENCODE projects for epigenetics and regulation, sort of ongoing efforts to create single-cell epigenomic data sets. And it was mentioned yesterday, obviously, the integration between imaging, spatially resolved transcriptomics, for instance, and other imaging methods, and HCA sort of single-cell RNA-seq data sets, for instance, would be very important, and obviously probably require separate portals for doing this. Again, I think the comment was made a couple of times about ease of use, that some of these portals would have to be very accessible to biologists and wide audience, while others would want to basically be accessible programmatically through APIs and other means. Okay, technical challenges. So I think half the audience we counted consists of people who would like to create portals, and so for them it's actually very important to have the right infrastructure and access to this data. So there's some discussion about just how to make sure that this could be run on a sort of cloud computation. Sorry, this is actually leftover from previous one. So a key point was that DCP, they provides infrastructure to access sort of low-level results. They're basically not trying to impose any sort of conditions or restrictions on the portal creators beyond that, and they're not trying to compete with portal creators, so there's a bit of a boundary that has to be drawn there. But other than that, basically input from the portal creators as to what API elements are needed, what access elements are needed, or what pre-computed results might be helpful in creating these portals is very important. And so kind of jumping ahead a little bit, this session was mostly to solicit community input, and we hope this will be sort of a starting point for doing this. And there are a few ways to continue this forward. So specifically for portal creators and the integration of DCP, coordination of DCP, so there's both a Slack channel called Tertiary Portals, and also there's a bi-weekly conference call that discusses these issues and access APIs in particular. So certainly for portal creators, I think it's an important resource. There was some discussion about file formats, being able to, again, agree on some formats that would be useful for a variety of portals, so they could be pre-computed and represented by, or served by DCP. Examples would be, for instance, whether we want to use something like HDF5 or some restricted version of it, what would be sort of additional requirements for these formats. Like HDF5 can be pretty hard to parse from some languages and can be fairly complicated. And if you keep it in general, for instance, there are some efforts to define, like Loom, to define sort of a restricted subset of HDF5 that would be easier to parse and easier to handle by a variety of packages. These are leftover points from. So let's see other other technical challenges. Right. I think that was it for technical challenges. Okay, so soliciting community input. So as I mentioned, the Slack channels and biweekly conference calls, we also created a Google Doc that was actually meant to be initially a summary of this discussion that we had. But we wanted to be kind of a living document, so we'll put up the URL here, and we would like people to basically contribute to it further, to describe both what they think are key sort of computational challenges in general in analysis of this data, but also technical things related to, let's say, data formats, APIs, and other bits that your DCP could help with, or we could agree on centrally. There's also a section there on on annotation that's quite extensive. Again, I think community input on cell ontologies would be quite important here, and how to coordinate this type of manual annotation. Thank you. That was it. Any questions?
Thanks, Peter. That was great. So we've got time for some questions, and there'll be some microphones running about. Please use them so we can get your questions on the live stream.
So this is actually not a question, but a comment to the audience. So we are really trying to think of how to make this data accessible and useful to biologists. And I can say from the whole group of us that you guys as biologists haven't been giving us enough input in how you want it, what's biologically meaningful, what's a meaningful cell type for you. And if we don't get this input, then it's going to be a very mathematical set of tools and a very mathematically defined set of cell types and definition for cell type, and it will be a very mathematical object. And the more input you give us, the more biological it will be.
I'm not sure incorporation with the input data is a good idea, because input data is mainly from the cell lines. They're not the primary cells. I think maybe a more appropriate project we want to incorporate it with, it's probably the roadmap of epigenomics. These are data from primary tissues.
Certainly. I just want to emphasize, these are this is supposed to be examples of integration that could be done. And again, it wouldn't be part of the core HCA framework, but one of the portals that could integrate. We were just trying to solicit use cases. And then I think to Dana's point, so one important piece of discussion. Oh, okay, please.
Well, yeah, as a user here, Dana, I really appreciate your input here. So what's the best approach to interact with your group? Because it's not necessarily clear from where I'm standing how we could best give you this feedback in an effective manner.
So Peter.
Okay. Well, so I think Dana's question was more general with our specific group. So again, we're soliciting input in this Google Doc that we'll eventually try to propagate into something more formal. But certainly input from biologists as to what level, let's say, of interpretation, annotation, tools they would like to see is extremely important at this point. I think, more general question as to how biologists can contribute to computational groups. Dana can have some ideas on this. I think we do have this analysis working group that's supposed to be listening to a wider audience, and certainly that would be one point of communication. I don't know if we have anything more systematic than that at this point.
Yeah. Okay.
Okay. I guess one kind of related point that I forgot to mention that they came up in the discussion, actually it was fairly heated discussion there, is again has to do with the level of interpretation that would be more suitable, let's say, to biologists versus computational people, and that was a use of latent variables. And so for some of these analysis results, right, for annotation, let's say evidence and so on, ideally we would like to see explanation in terms of simple, single genes or single markers, and a lot of times it may not be realistic. So I think that there's kind of a gradient going from single markers to extremely complex latent variables that were formulated using entire transcriptome, and there has to be some sort of middle ground there. And we started this discussion again. I think maybe some useful guidelines will come out of this that we can then implement in the algorithms.
The first thing is to have more biological presence when we break out for computation. So I've been trying to convince biologists to come to the computational breakouts, and at the end they become a bunch of computational people technically arguing with each other without enough biological input. Now, I don't know if there's quiet biologists in the room that hear the technical jargon and shut down and sort of zone out, or they just don't come in the room for the first place. But the more biological voice there will be in these breakouts, the more biologically relevant our end result will be. So there is just engaging more in these general meetings and not saying, oh, that's computation, that's not me. Because if the biologists don't come to the computational meetings, then breakouts, then their input won't be heard, and what will be the final output. And I think it's really critical, so this will be really both biologically relevant in terms of what we define and biologically useful so that you could use this and glean insights. So the more you, as biologists, give to the computational group, the more you will get back. And I really like engaging with people, so even though I'm a busy person, this type of thing bugged me.
Time for one more question or comment?
So I understand the importance of bringing the biologist to the table, but I think that we need to figure out how to not have our breakout discussions be about the micro issues. In our development session, we didn't talk about what forceps, pipette tips, cover slips, or microscope objectives we would use. And it sometimes seems when I sit in the computation sessions that it breaks down into implementation plans instead of goals. And I think we need to establish the goals first and make sure that we have a broad enough set of them that it really encompasses what all of us want, and then break out into the implementation. And I worry that in the computation sessions, sometimes we fall into, probably because we aren't having enough biologists in the room, into discussing the secondary things, the implementation, rather than the primary, setting the specifications of what the needs are.
Yeah, I think it's certainly appreciated, and the technical challenges sort of, I left as the last slide just because we have community input coordination here, but in general this is supposed to be a small section in there, but the other sections relate more to sort of either central, let's say, annotation issues for the atlas, or sort of computational tools that people would like to create, right? So certainly, you know, input at that level is most appreciated.
That wasn't so much a comment to your slides, which I thought were excellent, but the comment Dana was making, I'm assuming it's Dana because I can't see from behind here, about the need to get us at the table. And I agree totally. I just think that we should make sure that we're having separate sections for the two goals.
Okay.
So the elephant in the Slack channel here is between what Dana was saying and what the DCC sort of was presenting us as their end goal, at least for stage one portal development, is how much biology is going to be capturable from the portal outputs. And I guess what we were proposing through all this annotation discussion was that we get biology attached to clusters in data sets in a sufficiently informed way with evidence trails such that when a user can get an easy output download from the portal that says here's some as tagged cells that have these annotations, and you get class and subclass structure from the cell populations that are present that we can then take through other analyses, at any level, computationally or relatively manually.
Yeah, so certainly, I guess in the discussion it was pretty clear that the annotations will be one of the key vehicles for importing the knowledge of the biologists have and their opinions into HCA. And obviously we will continue to work on this to figure out what are the both user interfaces and the APIs and the sort of the infrastructure that's necessary for doing this, but it will be one of the key vehicles.
I think we're going to have to move on. Sten, if you could make a quick comment and then we'll move on and perhaps carry on this discussion over lunch.
Okay, I just want to say I think annotation is really a super important part of this project, and I think it should be separated from the data and to be an entity in itself. And the key challenge of annotation is to encode biological knowledge. So it's not the matter, the challenge is not how to, technically how to attach label to clusters. The challenge is to encode the biological knowledge that when I see this feature, it is an astrocyte or it's a cell in the cell cycle. And so that has to be a separate entity that we evolve and iterate and collaborate on and build as a body of knowledge. And then we can discuss how to apply that to cells and clusters and so on. That's a technical challenge. But the biological challenge is to encode our collective knowledge and the collective knowledge of biologists.
Thanks, Sten.